this is your revision summary for CB3, which is all about genetics. So we're going to be starting off with DNA. So DNA stands for deoxyribonucleic acid. How we find DNA in the nucleus is in chromosomes. So you normally see a chromosome like this because this is how it's copied. So it's easier to see them when they're being copied, and that is what gives us our X shape. So this here is a chromosome. A chromosome is a long strand of DNA wound around proteins. So long strand of DNA wound around protein. That means that we have chromosomes, which are long strands of DNA, which then hold our genes. Okay, so it's a string of genes, and genes code for proteins. So over here, I've drawn a structure of DNA. So what we need to know is DNA is a double helix. It's found in the nucleus. The backbone is phosphate and sugar. And then the backbone is joined by bases. So what that means is, if this is my strand of DNA, this line here is my backbone. That is my phosphate sugar. In the middle we have bases, okay, and between them they have bonds. So it's a double helix because it has two strands and they are twisted. It's easy to think about this as a ladder that is then twisted. So it is a twisted ladder. So each section, like this, is known as a nucleotide. Okay, so this is a nucleotide. So if we go down to a smaller level, we can then see this is our phosphate. This is our deoxyribose sugar. And this is our base. So that is one part of this. DNA is known as a polymer because it's made up of many of these. This is a monomer, it's one. A polymer is many of these put together. So DNA is a polymer because there's lots of nucleotides held together. So the DNA strand has base pairs. Specific bases go with specific bases. So A always goes with T and C always goes with G. So A and T, C and G always go together. They are complementary. And then, as you can see, between the bases, I've got these two dashy lines, and on this side, I've got three dashy lines. These are weak hydrogen bonds which hold it together. Okay? So, if we then have all of these bases in our DNA, they code for specific proteins. So we have this word down here called genome, which is the base sequences of all the DNA in an organism. So the base sequence of DNA in an organism. So the genome is all the base pairs, all the genes. The next part to think about is genes and alleles. So genes are short parts of DNA that code for specific characteristics. So a gene is a short piece of DNA which codes for a specific characteristic. An allele then is many different versions of the same gene. So an allele are different versions of a gene. So genes are the short part of DNA which codes for a specific characteristic, and then alleles are different versions of a gene. So what that means, if you've got your chromosomes that look like this, I've tried to pick specific points. These would be genes on a specific chromosome. If on the same chromosome they are the same, like here and here, they are called homozygous. So homozygous, homo for the same, and then if they are different, so if they have different alleles of the same gene, they're called heterozygous. So that is same allele on both chromosomes. Heterozygous means different allele on chromosomes. Bit to add there is we have 23 pairs of chromosomes in the human body. So the alleles are going to be on the same pair. So the chromosomes of the same pair have the same genes. So this is a chromosome of one pair. So chromosomes of the same pair have the same genes. What that might mean is chromosome 3, the pair for 3 may code for eye colour and hair colour. This bit here could be eye colour. If they are homozygous, they're both coding for the same gene. Whereas if this one down here is heterozygous and that's hair colour, they could both be coding for something different. So these are a pair of chromosomes that have the same genes. Practical that you can be asked about is extracting DNA from fruit. So what that looks like is you need to know the method. So one is grinding up the fruit using a pestle and mortar. So grind up fruit using pestle and mortar. What that is doing is it's separating the cells. Then number 2 is add detergent to break open the cell membranes. So add detergent to break open cell membrane. And then the last bit is adding ice cold ethanol, which causes DNA to precipitate. You can then take that precipitate and put it onto a slide and look at it under a microscope. It's now important to understand the different genetic definitions. Here I've got genotype. I have spoken about this before, but this is the genetic code of the individual. So genetic. And the phenotype are the characteristics that are seen. So it's the visible characteristics. So in this case, we've got lots of letters. We've got big B, big B, big B, little b, little b, little b. Big B is for brown. Little b is for black. So we're now talking about what characteristics are going to be seen if we have those alleles of the gene. So we talked about having the same allele as homozygous. So if we have big B, that is known as homozygous because they are the same. A capital letter also shows us that it's dominant. Here we've got big B, little b, so that means they are different. That shows that they are heterozygous because they have one of each allele. Now down here we've got two of the same allele, but they're both little letters. That means they're homozygous recessive. Okay, so we've got little letters for recessive genes and we've got big letters for dominant. So what that means is the recessive will only show if there are two copies present. Dominant will show if one copy is present. So what that means is this dominant one will always overshadow the recessive if one of them is there. So this big B is going to show brown. Okay, so that big B was for brown. Big B, little b is also going to be brown because we've got one copy, so it overshadows that black. Now, if we have little b, we get black because we have two copies present of these little ones. So dominant is these two here. So the visible characteristics that will be seen from our genotype of big B will be brown hair colour, and if we have big B, we're going to have brown hair colour. Little b, we get black, so that is recessive. Now we're going to be looking at monohybrid inheritance. So that is this kind of diagram here which shows us what possibilities we can have for the offspring and their genetics. So this is the mother, this is the father, this is their gametes. So we only have half the number of chromosomes in each gamete, so they get one of each chromosome that meet up with one from the mother and one from the father. So that's what brings us this bit down here. So this is the possible genotype of the offspring. So if we remember, big B was brown and little b was black, we can see that we've got brown, 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 black. So this then is the phenotype. So this is the phenotype, the characteristic that's seen. This is the genotype. From this diagram we can see the chance of a child having brown hair or black hair. If we have a look, we have 1, 2, 3 that have brown, so that means it's 75% chance of brown and 25% black. We can do that because we know there's 4 of them, and if you divide 100 by 4 you get 25, so each one is worth 25%. You can also put it as a fraction, so you could have 3/4. You could also say 3 out of 4 or 1 out of 4. But that is how this diagram works. You pull your gametes from your mother and your father down so that they meet and breed. So down and meet with the father, this one down here and meets with the father. So that's how you get your genotype of your offspring from the gametes. And then your phenotype is the characteristic that is visible. Another way of finding out whether the percentage chance of someone being brown-haired, blue-eyed, male, female is to use a Punnett square. So we can use these squares that look like this to work that out. First of all, we're going to do sex. So if we have a male and a female mating, a female is going to be XX, so this is female, and a male is XY. So here I've got a female, so this is the mother, and this bit here is the father. So when they mate, they have XX here, so you just put into the box what you see. So XX, XX here, XY, XY. So that means that they have 50% chance of being male and 50% chance of being female. How I can work that out is each box is worth 25%, so 25, 25, that's 50 for a female, and 25 and 25, that's 50 for a male. They are determined by one pair of sex chromosomes. So sex is determined by one pair of sex chromosomes. Another example of using a Punnett square is with this WW and small w's. So if we say that the big W is dominant, as it is, so big W is dominant, little w is recessive. So here in this box we get big W, big W, big W, big W. Now we've got big W and little w, the big W always goes first. So big W first, little w second. So that means we've still got 50% chance. Oh no, hold on, we haven't. We've got 50% of these have homozygous dominant. 50% of these are heterozygous, but all of them, 100%, have a big W. That means that the big W will always be seen. So it's 100% dominant. So big W seen. So the next part to talk about is variation amongst people. So variation is all the differences in a population. Okay, so variation is all the differences in our population. That can be caused by either environmental or genetics or both. So if it's genetics, it's caused by the genes that you inherit. If it's environmental, it's caused by the surroundings, okay? So caused by different conditions. So let me use a different coloured pen. If I'm thinking about genetics, this is going to be things like hair colour, eye colour, blood type, and skin tone. Now, if it's environmental, this is going to be things like scars, hairstyle, tattoo, suntan. But these things can be done by both. So what that looks like is caused by a combination of environment and genes. So that could be something like weight or height, as you have specific genes for these things, but the amount of food and nutrients will also affect how heavy you are and how much you grow. There are two more parts to variation. We've got this continuous and discontinuous. So we can put our variation into two different categories. So continuous means that it happens over a range. So this is going to be things like weight, height, and handspan. Whereas discontinuous are things where they go into a specific group. So they are into specific groups. That means things like blood type, eye colour, hair colour. The Human Genome Project is a really important study which has been a collaboration between lots of different scientists to decode the human genome. It was started in 2003, but it's particularly important as medicine moves forward so that people can see the genome of different people so that we can move towards personalised medicines. Now that means that people will be able to have their genomes read. They'll be able to see what diseases they may get in the future, what genetic problems they may have, what recessive genes they may have, what dominant genes they may have, but also it allows doctors to tell you what medicines will work better for that specific person because of their genes. Last and probably very challenging part to this module is meiosis. Now it's very similar to mitosis that was seen in CB2, but there is an extra added stage. So meiosis is all about making gametes. So we're taking this diploid cell and we're making them into haploid cells. So what happens then if we've got our set of chromosomes, and this is our parent cell. Our parent cell is diploid, so it's got two sets of chromosomes. It goes through the first round of PMAT. So this is how I recognise it. So that's prophase, metaphase, anaphase, telophase. That then gives us these two cells here. And what's going to happen then is the parent cell divides and each chromosome is copied. Then it's going to divide again. So this is our first round, and our parent cell divides each chromosome is copied. And then we get a second round of division. So we get this PMAT again. So PMAT, prophase, metaphase, anaphase and telophase. And that gives us 4 non-identical haploid cells. So each daughter cell gets a copy of a chromosome from each pair. Each daughter cell has a set of chromosomes, so they have one set of chromosomes. So at this point they only have 23 chromosomes, not 46. Okay, so this is division again. So PMAT will also be known as division. So with meiosis then, when I've written PMAT in both places, we've got 2 rounds of division. That means we've got prophase, metaphase, anaphase and telophase. Okay, so these cells at the end are our gametes, they're our sex cells. So we've got our sperm and our egg. So really quickly then, if we have a parent cell, that copies and divides itself into 2 duplicate cells. So it's got exactly the same as the first cell, but it's got 2 copies. It then duplicates its chromosomes again and splits into 4 with half the number of chromosomes. We get genetic variation from this process, as sometimes when these line up, the chromosomes can cross over and they swap parts of their chromosomes with one another. It's also random assortment in the way that they line up and the way that they are pulled apart, and the way that they are then put into their final cells.